with 3 days before the seminar, what do? We have no seminar speaker. Well, the times sort of require us to improvise, and I'm going to be teaching virology this afternoon, molecular virology this afternoon, and I was going to give an overview of what we know about the coronavirus. So I thought that I would adapt that lecture to present to the faculty. And that's what I'm going to do today. So I guess I've got a number of messages during this talk. Before I start though, I want to thank all those people who sent me material. I didn't have time to read everything, and that's also going to be one of my things that I want to say up front, is that I'm not a coronavirus expert. I'm not an animal virus or human virus expert. I work with viruses of bacteria, cyanobacteria in the marine environment. But I think all of us have done some reading about this virus and about this epidemic or pandemic. And what I'm going to try to do in this talk is to give more of an overview, together with Yoram, of what we know about this virus, and also a little bit about what we don't know about this virus. And one of my take-home messages is that we really need to take this virus very seriously for what it's doing. But I do want to emphasize that this should be less for for fear of our personal safety for most people, and more for the safety of the community at large, in particular those that are compromised within the community. Okay, so it doesn't just, oh boy.
Maybe a question, maybe?
Sure.
Where will we be able to find the recorded talk if we would like to?
I don't know. I don't know. Once I work it out, once I get the first one, I'll work it out and I'll let you know. Okay, so let's start. So what I'm going to do is give an introduction to the whole situation. Then I'm going to talk a little bit about who are the human coronaviruses, because this isn't the first human coronavirus that is known. Then I'll give an overview of the infection process, a bit of the biology behind what is known about coronaviruses, but this is not specific, most of this is not specific to the coronavirus that is causing the present pandemic, but we're assuming that a lot of what goes on will be similar. Then I'll talk about the evidence that exists and is fairly strong at this point in time for the source of the SARS-CoV virus. Then talk a little bit about the epidemiology of COVID-19 disease, a bit about the prospects for solutions, and what's being done around the world and in Israel. And then Yoram will take over and talk about the progression of the disease, and also the work that's being done to develop vaccines, and then I'll sum up. So this is an emerging disease, and the relevance of it being an emerging disease is that this virus has come into a naive population of humans worldwide. And a naive population basically means that we do not have immunity as a population and personally. And this is one of the reasons why viruses that are emerging into the human population for the first time can cause such major havoc. The virus that we're talking about is called SARS-CoV-2, being called that by the WHO. It stands for Severe Acute Respiratory Syndrome Covirus 2, because it's the second one to cause similar harsh symptoms. And the disease has been named COVID-19. Sorry, just playing around with the temperature in my room here. Okay, so if we have a look at the global distribution of COVID-19, we see that over 150 countries have been affected so far. And this is actually from 2 days ago, and things change so rapidly, it's probably more countries. The countries that aren't affected are those that are in grey here. All other countries have been affected, have found instances of this. And the number of instances that have been detected you can see the legend here. But as of 2 days ago, there was over 300,000 cases that had been detected, 13,000 deaths worldwide, and about 95,000 people who've recovered, which basically means we have another 200,000 people that have neither died or recovered and will go into one of these boxes. When we look at the number of cases with date, and this is from the 31st of December until the 21st of March, it's under 3 months. This pandemic has been going on for under 3 months. What we can see here is that the initial outbreak and instances occurred in China, and that through measures that they have instituted, the problem has reduced dramatically, and it is under control in China at present. However, it is then turned and moved into the rest of the world. And the rest of the world is mainly, at this point in time, Europe, Italy, Spain, France in particular, the US in orange, and Asia, other places besides China, fairly stable numbers, okay? So this is a bit of a timeline with somewhat of an Israeli emphasis. First clinical case was hospitalized in Wuhan on the 12th of December. It was reported as an outbreak on the 31st of December by China to the World Health Organization, which is probably 2 weeks too late. It also happened around the time in China where there was a holiday and people traveled from Wuhan, from where it started, to the rest of China in massive numbers, and also overseas. The first death was reported in China on the 11th of January, and it first appeared in Europe, was first detected in Europe on the 24th of January in France. The World Health Organization declared a public health emergency of international concern at the end of January and instigated all sorts of recommendations and sending of materials to the world to prepare for the problem. The first case was detected in Israel on the 21st of February from somebody coming back from, I can't remember if it was Greece or Italy at this point. The first transmission in Israel was detected on the 1st of March between a worker and between two workers, between a man who'd come back from overseas and his worker. On the 11th of March, Israel started putting in limitations on gatherings. Initially, before that, we were trying to contain the problem by preventing people who came in with it from spreading it. But by 11th of March, it was pretty clear that containment wasn't working, and we were moving into what's considered the mitigation stage, where is to do as much as possible to prevent spread within the country. On the 11th of March, the World Health Organization declared COVID-19 a pandemic. Perhaps I should have had those two things switched in their order. The first death, unfortunately, occurred in Israel on the 20th of March. Now, using what we know now about clinical indications, and also based on diversity of the virus looked at through genome analyses, we can assume that the jump into humans occurred in late November, early December, which goes together with the 12th of December first clinical indication of it. Okay, so now I'm going to go into a little bit about what this family of viruses are. So these are coronaviruses, the order Nidovirales, family Coronaviridae. Corona comes from the word crown or halo in Latin. These viruses are single-stranded positive strand genomes, have single-stranded positive strand genomes. The genomes are very large for an RNA virus, between 27 and 32 kilobase, and I'll come back to that further on. These viruses are enveloped. They have a lipid membrane surrounding them. They're in shape, they're either spherical or pleomorphic. Pleomorphic means that they don't have one shape, that their shape's sort of a bit flexible. And their diameter, their size is between 80 to 120 nanometers. And the nucleocapsid has helical symmetry. I suggest the reading of these two reviews for people who are interested in understanding a fair amount about their biology. I think they give a very good indication. Okay, so the coronaviruses that infect humans can be broken into two groups. 4 that cause mild cold-like symptoms, and it's said that anything between 10 to 25% of the common cold is caused by a coronavirus. Upper respiratory, mild disease. But there are three coronaviruses that cause more serious diseases. These are SARS, the first one was detected first in 2002, MERS that came out in 2012, now SARS-CoV-2. Okay, let's mute everybody. Okay. I thought I'd muted everybody. Whoever's got a music playing in the background, can you mute your sound, please? Okay. And so these, and the second one is the Middle East respiratory syndrome that came out in Saudi Arabia, and now SARS-CoV-2, both SARS-CoV and SARS-CoV-2 originated in China. Okay, so if we have a look at the genomes of the coronaviruses, they're fairly, I would say even very conserved, as we know for families of viruses. The genome consists, 2/3 of the genome, this isn't drawn to scale, 2/3 of the genome is made up of 2 open reading frames that code for the replicase, which is a group of proteins, includes proteases, includes a single-stranded binding protein, an RNA-dependent RNA polymerase, helicase, and an exonuclease that I will also come back to later on in the talk. The rest of the genome is made up of structural proteins, the spike glycoprotein, which is involved in recognition and entry, envelope protein, a membrane protein that is involved in assembly and exit, nucleocapsid protein that makes up the helical protein part of the nucleocapsid, and there are also some accessory proteins that I'm not going to go into at all. Now, what I'm going to be talking to you about now is stuff that is known for other coronaviruses and not for SARS-CoV-2. All right, as with any virus, the ability of the virus to recognize the host determines the host range and tissue tropism. In other words, what tissue types the virus will infect. The spike glycoprotein in coronaviruses is the protein responsible for recognition and attachment to the human receptor, cell receptor, as well as entry. And so first, this spike protein has 2 domains. The S1 domain, the bulb part, is what interacts with the human receptor and leads to endocytosis. Within the endosome, there is protease activity that prepares the spike protein, cleaves between, I don't think I showed you, between the S1 and S2 part. Okay, they don't show it here. I'll show it later then. Between the S1 and S2 part, and prepares or activates the S2 stalk part of the spike protein to fuse the viral membrane with the endosome membrane. This leads to uncoating and release of the viral genome into the cell, and then it can be replicated and transcribed. So the expression of the genome occurs in two stages. Since this is a single-stranded RNA virus of positive strand, it in itself directly can be translated, and the first only part, or the two-third part encoded by ORF1A and ORF1B, is translated immediately after entry into the cell. This produces a polyprotein or polyproteins, first a polyprotein of 1A, and then through a -1 frame shift, a polyprotein of 1A and 1B. And these are the proteins that are produced, including two proteases that are involved, although may not be the only proteases, involved in breaking this polyprotein into functional proteins. One of the proteins in the replicase set of proteins is an exonuclease. This exonuclease was shown in 2011 by Dennison et al. To have proofreading capabilities. And basically what they did was they inactivated the exonuclease active site and had a look at mutations that occurred in two different viruses. And what they found was that the inactivation of the exon protein led to 15 to 20-fold more mutations. Okay, now why I'm telling you this is that RNA viruses in particular are known to be highly mutated and very variable. And this is one of the reasons, not the only reason, one of the reasons that each year we need a new influenza vaccine is because these viruses change so much. We don't know yet for sure that this covirus 2 has this ability. We assume it does based on, right? I assume it does, I should say, based on what's known in SARS-CoV-1. But if this is the case, then it's possible that a vaccine, once it is developed, will provide immunity to this virus in a long-term fashion. It's still, and the reason that they have no proofread RNA viruses mutate so much is that RNA-directed RNA polymerases don't have proofreading abilities themselves, and this is a protein that provides that to this virus family. Okay, after uncoating, we have, as I said, direct translation of the polyprotein 1A and 1B that produces the replicase. The replicase machinery then produces a -strand full-length genome copy of the genome, which is then used as a template to produce the +strand whole genome length genome that is then later on packaged into the nucleocapsid. This happens in a replication transcription complex, which is membrane bounded. The replicase also transcribes subgenomic parts of the genome for the for the structural parts and ancillary parts of the genome, structural proteins that make the actual virion and membrane proteins, etc. The N protein, the nucleic acid protein, is thought, it's not known yet, thought to be involved in the switch between producing whole genomes and producing subgenome particles. Okay, once we have full genomes and the nucleocapsid protein, we get the nucleocapsid protein interacting with the genome and forming a helical structure around it where the RNA is physically interacting with the N protein. There is formation on the endoplasmatic reticulum of a number of the glycoproteins. There is a recruitment of the nucleocapsid and the membrane protein in the ER Golgi intermediate compartment, where we get all of the assembly happening and formation of a vesicle that is then budded out by exocytosis to the extracellular region before going into another cell. Okay. All right, so that's the infection process. Now let's talk about SARS-CoV-2 and the evidence that exists for its origin. Where did this virus come from? Where did it come from to infect humans? So genomic analysis that was done by Zhao et al. I've written, by the way, for the papers that are relevant, the actual date when the papers came out, because sometimes that is very relevant for what is known, because something that came out 2 weeks earlier or even a week earlier can be significantly updated within a week. The publication of data on this is extremely rapid. I mean on the virus in general. So sequencing of this genome of 5 different isolates from 6 different people, 5 different people, showed that, first of all, those isolates from 5 different people early on in the disease were very similar to each other. These genomes were only 76 to 79% identical to SARS-CoV virus. I've heard the number 76 thrown around. So it looks like the origin is not the original SARS-CoV virus that could infect humans already. And it was found to be 96% identical across the genome to a bat virus that's called RA-TG13. And RA is for the name of the bat species, and I don't remember what it is right now. What we see here is the structure of the genome. The organization is similar to those known for other coronaviruses, and this is the sequence identity to, in red, to the SARS virus that infects humans. In blue here, to the rat virus, to the bat virus, and in green and red to other, in green and pink to other bat viruses. We have a look at the phylogeny based on the whole genome of this virus. We see that this is the cluster of these human coronaviruses, and it clusters very closely to this bat coronavirus, and separately from the SARS virus and the MERS virus that infects humans. So this has led people to say that it is most likely this virus jumped from bats into humans and wasn't of human source. And this research is newer than the research that suggested that this came from a pangolin, an armadillo type organism. because that original paper only looked at the spike protein. If we look at the spike protein, we see it's the most diverse part of the genome, or one of the most diverse parts of the genome, and that's to be expected. That's the region that attaches to host cells and enters into it, and that's under a lot of selective pressure as resistance develops, etc. I'm not going to go into that too much, but now looking at the diversity in the spike protein can tell us quite a bit about what might be happening with this virus relative to other viruses. So this is the spike protein. It's got two subunits, the S. Ooh, can people hear me? I've been told that my internet access is not stable. Can you put thumbs up if you hear me?
Can Shomin.
Okay. Okay, so S1 subunit that is responsible for recognition has 6 amino acid differences relative to the SARS-CoV virus. In addition, the region between the S1 subunit and the S2 subunit, there was a 12 nucleotide insertion that introduces a furin cleavage site. These two things are things that I will come back to in a moment, and may be very, very relevant for how this virus differs at one level to the SARS-CoV-1. Okay, so a number of studies have been done to indicate that ACE2 is the receptor for SARS-CoV-2 as it is for SARS-CoV-1. This is an exopeptidase that cleaves angiotensin 1 and 2 and leads to vasodilation. What this particular study did was they expressed ACE2 in HeLa cells and looked at the ability of different spike proteins to, different viruses to enter into these cells. And what they found was that the human coronavirus, sorry, I'm getting this wrong. These are ACE2 proteins from different organisms: human, bat, swine, civet, mouse, and HeLa cells that were not transfected with anything. What we can see is that the virus, the coronavirus 2, can enter and infect humans, bats, swine, and civets, but not mice. So it has a fairly broad host range, at least on entry and infection. And it when it's not transfected with any of these, then there is no infection either. So this suggests that this is the receptor. There's other studies that also suggest that this is the receptor, and there's also been work done by chemical work that shows that the spike protein from CoVirus2 has a higher affinity to ACE2 than does CoVirus1. The implications for this is that perhaps, and this is an assumption at this point in time, perhaps less virus is needed to initiate an infection. Okay, so the spike protein has two parts. We talked about that. S1 for receptor recognition, and S2 for fusion between the viral membrane and the host membrane. So a host protease is known to be the one that activates the spike protein for this fusion. The question at this point in time is which host protease does that? There are a number of, at least two differing views at this point in time. Anderson in 2020 that detected this 12 nucleotide insertion that introduces a furin cleavage site suggests that furin is involved, as is known for MERS. And this is partly due to work that has been shown that when you insert this nucleotide between S1 and S2 in covirus 1, you get enhanced fusion without affecting entry. There's another study by Hoffman et al. In 2020 in Cell, February, that says that this cleavage is probably by TMPRSS2, as found for SARS covirus 1, because inhibitor of this protease blocked entry of SARS covirus 2, and also immunological sera from SARS patients neutralized and prevented SARS-CoV-2 entry. What perhaps is what is going on is perhaps furin precleaves the S1, S2 protein prior to TMPRSS2 cleavage, as suggested by Hoffman, but the jury's still out exactly on what's happening there. Okay. Transmission. Now getting to a bit of epidemiology about this. Transmission depends on a number of factors. Transmission, I should just define, transmission basically means the movement from one infected organism to another infected organism, or one infected person to another infected person. It depends greatly on the concentration of released particles. In theory, one virus can infect a cell, but in multi-cell organisms, one virus won't reach a cell because we've got many, many layers of defense, including our skin, mucosal linings, single individual cell protection, including apoptosis, etc. So the number of viruses that reach an organism are relevant on how many of those viruses reach a cell that has a receptor it can recognize. How well transition works also depends on its ability to survive in the environment. The effectivity of the virus to recognize and attach, as we talked about just previously, and the effectivity of host defenses throughout the scale, from skin, mucosal linings, apoptosis, and other single cell mechanisms, and the immune system. For instance, one thing about the environment, I'll get back to this in a bit, aerosol dispersed viruses, which this is expected to be, need to remain hydrated as they're inactivated by dryness. So shedding and transmission of respiratory diseases from the nose, mouth, and the size of the droplets that are released from our nose and our mouth, both when we speak and sing and lecture and sneeze and cough, is such that most of these droplets will drop down to the ground within 2 meters. And this is the reason for the directive to maintain a 2 meter distance from other people. So it's both airborne, and when these droplets drop on surfaces and are then touched by another person. So research has been done already, and people have probably heard about this, about the stability of the virus in the environment. And what we're hearing in the literature is things like, what we're hearing in the news is things like more than 3 hours in air, not so long on copper, a day on cardboard, 3 days on stainless steel and plastic. This is true. You can find infective viruses at that level. But have a look at the decline in the virus titer during the period of time that those research things are done, and that is not being discussed in the literature. And taking that together with you need more viruses to be infected. If you if you touch a virus that's been on stainless steel for 2.5 days, your chances of being infected by it are much less than if it's been there for a couple of hours. But we can see that the stability of the virus declines with time, and it's different whether it's in the air in aerosols or on different surfaces. Okay, there's been a fair amount of discussion about seasonality. Is this virus going to be seasonal? Perhaps. We don't know, and we won't know until it happens. Many of these viruses, both influenza viruses as well as coronaviruses, have a seasonal pattern of infection. Much higher in the winter, much lower in the summer. Higher in the winter due to a number of factors. There are a number of hypotheses. One of the most convincing hypotheses is related to temperature and humidity. Low humidity, low temperature keeps the droplets, keeps water droplets in the air for longer. And this is work that was done on influenza. Sorry, I couldn't find the original paper. If somebody's interested, we can look for it. But it shows that at 5 degrees, virus particles are stable in nuclei which are less than 5 microns in size, in aerosols. Stable, and there's 100% transmission. There's good level of transmission. And that with increasing humidity, even at 5 degrees, we get a decline in transmission. At 20 degrees and low humidity, we have good transmission. At 20 degrees and increased relative humidity, we have dropping out of the droplets. And at 80 degrees humidity, they're dropped out entirely. So this might happen. If this does happen, I think we can probably expect, for this particular virus, this might happen for this particular virus, we can expect a drop. I don't think we're going to come down to 0 in the summer because of the way this virus is transmitted. And if it does, chances are we'll get resurgence again in the fall. Something to keep into consideration. Other hypotheses as to why respiratory viruses are seasonal. It's got to do with mucosal surfaces in our bodies and our immune system. I have no idea how scientifically based those hypotheses are. Okay, a little bit more about the epidemiology of this particular virus. There's a term that's called the reproduction number, R0 or R0, which I think probably everybody's heard about. This number relates to the average number of people a single infected person goes on to transmit the disease to in a susceptible population. If you've got an entirely susceptible population, there's a set value if nothing is done. But distancing people from each other, preventing droplets reaching other people, can change or reduce the effective reproduction number or transition to lower values. When you have transition values greater than 1, in other words, on average, on a population level, each person infects more than one person, you have spread and an increase in the disease and can potentially cause an epidemic. The estimates for SARS-CoV-2 range, and they range because we're in the middle of an epidemic or a pandemic, and the real numbers are changing all the time because it depends on how you detect who's infected. Probably the most accurate number convincing study that I've heard about is one by Neil Ferguson and Imperial College, where they looked at data where everybody coming off planes from Wuhan early on and going to Europe were tested for the presence of the virus, irrespective of whether they showed symptoms. And based on that, they did some models and suggested that the transition number is 2.9. Most estimates range between 2.2 to 4.1. These are massive. To compare to seasonal flu, the R0 is 1.3 to 1.5. These numbers, on one hand, might be might actually be low because we don't know how many asymptomatic people this includes, except for the Wuhan outbreak. And there is a little bit of evidence to suggest that the one coming from Italy might transmit better. Case fatality rate is a measure that tells us how many people die out of those people that were reported as having been infected. It's different to a fatality rate of infections, because the fatality rate of infections requires that we know how many people are infected, and we're far from knowing that at this point in time. We can only do it based on who has been, who we know has been infected, and that depends on testing and symptoms. But this measures the proportion of detected deaths, detected cases that cause death, and it's very difficult to accurately determine during an epidemic and pandemic, because it depends on who you test, how broadly you test. The more people you test, the more asymptomatic people you include, that number will decline, on the one hand. On the other hand, this virus takes about 2 weeks, between 5 days and 2 weeks, to cause symptoms. So you need to accurately look at the number of deaths and compare that to the number of people that are infected. You have to take into consideration that lag period between infection and either death or recovery. So the 200,000 people we have that have neither recovered or have died, that's one of the reasons that that number is reported, because those are the ones we still don't know what their fate is going to be. So if we look at the percent death rates at 1 day to the number of infections at that same time, we're underestimating, because we should actually be comparing it to the number of deaths about 2 weeks beforehand. Okay, so the range of the estimates for SARS-CoV-2 is 3 to 4%, which is very high. But if we look on a per country level, it ranges between 0.3% and 8%. 0.3% is Germany. They measure, they test a lot of people. So perhaps this is low, but this number is still based on what is done, the number of deaths and the number of infected people at the same time, and not with the lag taken into consideration. So it could be related to two things. One, the number of people that they test, and two, healthcare, because the better the care, the lower the fatality rate is. And this is very clear from what's happening in Italy, where we've got an overall average of 3 to 4% infection globally. Italy has 8% mortality. Italy has 8% mortality because their healthcare has crashed. They're in a catastrophic situation, so we've got a very, very high level of mortality in Italy. Okay, so but once again, it's very important for me to state that these numbers change in the literature and in the news all the time, because we're working in current state as the pandemic is unfolding, and these values will only be accurate once things have settled down and have happened, either ended or recur over the years. Okay, so timeline of infection. Infection to symptoms is an average of 5.2 days. Symptoms to diagnosis afterwards is an average of 2.3 days. We have mild and sick people for up to 14 days, and more severe cases can last for up to a month. Infective period is still unclear, but it appears to begin at about 2 days after infection until many days post symptoms, up to a week or 9 days even, in one case that I read about. And what's relevant here is that the infective period of this virus is prior to the development of symptoms. And this is a highly dangerous disease to people with pre-existing conditions such as respiratory diseases, cardiovascular diseases, diabetes, hypertension, and cancer. For the rest of the community, it is less problematic. And this is broken down based on age, which I'm sure you've all heard on from the news. Okay, so let's compare, before I hand over to Yoram Reiter in another couple of slides, let's compare the epidemiology of SARS-CoV-2, COVID-19, to SARS and MERS, the other coronaviruses that we know. SARS occurred in November 2002 and pretty much ended in July 2003, originated in China. The source was from bats via palm civet. It underwent evolution in palm civet. It infected 8,000 people and killed 774 people. Transmission value estimated at 2 to 4. Mortality rate, fatality rate, close to 10%. Number of countries that were infected or affected were 29. MERS emerged in 2012 in Saudi Arabia. Source was for bats, seems to have undergone evolution in camels, infected about 2,500 people, killed about 850. Transmission in Saudi Arabia was less than 1, so it didn't go crazy. It was another outbreak in 2015, a small outbreak in South Korea that had an estimated higher transmission rate. Mortality of MERS was extremely high, 34%. That's massive. The number of countries affected were 28. If we now compare COVID-19 or SARS-CoV-2, emerged 2019 until we don't know when. China, probably bats was the source. Infected, I think I updated these yesterday, over 320,000 people known, confirmed to be infected worldwide. So far, 13,600 people have been killed. The estimated transmission ranges, but probably around 2.9. Mortality between also ranges, but probably overall, globally 3 to 4%. Number of countries affected, over 160 countries. So if we have a look at the transmission rate of SARS-CoV-2 and the transmission rate of SARS-CoV-1, fits right in there. Why is COVID-2 causing way more problems than SARS? Okay, so SARS was controlled by identifying cases and isolating them. This was doable, feasible, and done because people were infected when they had SARS, when they showed symptoms. COVID-19 is very transmissible and people can transmit. They are infective prior to them knowing that they have the virus, prior to them showing any symptoms. And it seems, it's not been proven yet, it seems also that there are many asymptomatic people that can also transmit the virus. So what this means is that this virus can be transmitted before you know you've got the disease. Therefore, isolating only people who have been proved to be infected is not so effective because you have a whole group reservoir of the virus in people who don't know they're sick, don't know yet or don't know at all. And this is what's causing the massive problems in Israel and all around the world in mitigating this pandemic. So mitigation is not sufficient. So what are the solutions? First of all, the solution that is being used all around the world, it's the only one that can be implemented immediately, is social distancing with the aim, in fact, all of the aims, to reduce transmission to below 1. When you reduce transmission to below 1, you reduce the spread and don't increase the spread. So if there's a transmission of 2.9, we need to reduce transmission 3-fold. So social distancing is what's being done nearly all around the world, if not all around the world. I can perhaps tell you that a study done by Chi-Hong Lin from Harvard Medical School showed that the initial outbreak in China had a R0 of 3.9. Social distancing reduced the R0 to 1.25. Still above 1, it's not sufficient. China then introduced centralized quarantine, putting sick people, mildly sick people, in community hospitals and hotels and stuff like that, and taking their healthcare workers and putting them in other homes, not with their families, because the health workers are one of the highly hit people. And then introduced massive screening for anybody who was contacted by somebody who was sick, and not only when they showed symptoms. And this brought the R0 down to 0.3 and is what enabled China to control this pandemic. Just to note that these numbers are a little different to a publication in Lancet. The Lancet publication, it was earlier than this one, and I think that these data are probably more accurate. Okay, the other things that obviously we want to be able to do is to find effective medicines to treat this. Existing antivirals have been tested. There's been anecdotal evidence that some of them work, but no clinical trials yet that show that this is real, and it's not very sure that there's anything that will work in general. Antibodies as a medicine is something that's being looked at in the Netherlands, Euramus, I can't remember the name of the company, where they've got an antibody. They've shown they've got an antibody that interacts with SARS-CoV-2. They would need to, they haven't undergone any human trials yet. They would need to produce massive amounts of the antibody to be able to use as a medicine. And of course, developing vaccines, which is obviously where we want to go. The problem here is, even though we have a number of people, academic institutes include, as well as private enterprises that say we have a vaccine that can work, they have yet to be tested and shown to be proven to be safe for us. If we use, Yoram will go into this in a moment, a lot more than I'm going to. I'm just going to mention that a vaccine, if it's not tested, it's known, it's been shown in cases that a vaccine can be more dangerous than the vaccine itself, and it has to be tested and shown to be safe before it can be given to people. Okay, so now I'm going to turn it over to Yoram. You can unmute your microphone and take it from here.
So hi everybody. First of all, thank you very much, Debbie, for the great lecture. So I'll start with a brief introduction, how the immune system is involved in viral infection. Of course, we have a massive involvement of the immune system, the adaptive and the innate immune system in the situations that we have here. And I'll try to explain what happens in a normal condition and why we see in a certain population, mainly the elderly people, why we see reactions that actually cause what we call massive or ARDS, which is acute respiratory distress syndrome that actually destroys the normal function of the lungs. So first of all, we have two systems that are involved in this story. So Debbie, it's not the order that I sent you. The slide.
Yes, it is the order you sent me. Do you want the next slide?
Well, okay, this slide I missed. Okay, so first of all, there are two systems that are involved in immunity, and we'll come back to it in more depth. But we have two systems. One is innate immune system that is associated with viral recognition, and this is the first line of defense that we have in our body to actually recognize and generate immunity against viruses. And then at the second stage, there is an adaptive immunity system that actually develops what basically eventually will protect us in the long term, and these are antibodies and T cells that actually can destroy or neutralize the virus. The problem is that what we are facing now is more a disease that is associated with innate immunity, because if you were exposed to the virus, you initiate innate immunity, which is mild and enables the adaptive immunity system to be initiated. Then actually you generate immunity, you have neutralizing antibodies, and you are protected. So even populations like young people that don't have any symptoms, actually they are exposed to the virus, of course, they actually generate a normal immune system even without any symptoms, and eventually they are protected. We'll see later that the major problem arises in people that something went wrong in their immune innate system. Actually, it is a balance between superactivation and also measures or mechanisms of the virus to evade the immune system by specific molecules and genes that we don't have time to go into it, but actually antagonize innate activity. So what you see in the slide here is actually the basics of innate immunity and the connection or the correlation and the interaction between the innate immunity and the adaptive immunity. You can see on the left side that basically the first stage of actually virus recognized by immune cells is what we call pattern recognition receptors. These are receptors that actually initiate the innate immunity, and they generate a response which is mainly associated with induction of inflammatory cytokines, such as IL-1, IL-6, IL-8, and others. And these cytokines actually protect us by their critical role in helping the adaptive immune system to generate at a later stage plasma cells that produce antibodies and maturing T cells that actually eventually will become effector T cells that will kill viral infected cells. What is happening in ARDS is also associated with induction of a super inflammatory cytokine response in which many of these cytokines that are being induced are actually expressed, are produced and function at such potency that actually they cause damage to the lungs, and this is mainly the problem in this patient, and this is actually what COVID-19 may cause. The most problematic, the things that we fear, and the fear of the healthcare system is that the elderly people will develop massive numbers of ARDS patients, which actually initiate pulmonary insults leading to disruption of the capillary activity and basically breakdown of the normal function of the lung, and this is why we need to go into intensive care. The virus actually affects a certain type of cells that exist in the alveolar, small, tiny roots of the lung, and they are also tropic specifically to a type of cells called the type 2 pneumocytes. These are cells that actually underlie the 5% of the alveolar surface, and this is why this tropism of the virus is so detrimental to people that have such an inflammatory response, and they enter into this ARDS condition. So in the next slide, Debbie.
Yoram, you're right. I put them in the other order. I'm sorry.
Yes, because I'm okay, but it doesn't matter. So if you go, well, it depends how much time we want to spend. On the biology and immunology. I think you can go for a moment for the slides. Do you have the slides on the innate system?
Maybe they're hidden. Maybe those slides were hidden. No. Oi, 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 oi.
Okay, doesn't matter.
Yoram, you can share your presentation from your computer.
Oi, oi, oi. Yes, why don't you do that? I can find what you sent me. Seems I just didn't copy them over.
Because it will, I will need time because.
So why don't you continue while I find them?
Okay, the presentation
I
generated on my.
Okay.
Okay.
I'll find them.
Okay. So, so what is important to I think as a basic information is that why do we have two actually systems that are working here? The innate immune system signaling is the earliest system that actually differentiate and recognize pathogen from cellular molecules, because usually we generate immune response against pathogens that are actually, let's say, not as pathogenic as viruses, and we have the adaptive immune system or enough time. Oh, here is the presentation.
Where do you want to go?
So in this slide, you can just see the type 2. Go to the previous slide.
Ah.
It's okay. I think nobody is rushing. Everybody has time.
I actually have to teach at 2:30, but I can't. No,
okay. So I'll finish in 10 minutes.
I can't get back the screen where I can move the slides back. Is this back or forwards? Shoes.
Okay, doesn't matter. Doesn't matter.
Here we go.
No, you went farther. Go one to the top. Oh, okay. Oops.
This one?
No. Go to the beginning of the presentation, and I'll tell you when to move to the next slide. Go to the first slide. Yes. Yes, this one. You want to put on presentation mode? Oh, wait. Okay. So here you can just see what I was describing before. You can see on the right panel, you can see the schematics of the alveolar cells, and you can see type 1 and type 2 pneumocytes, and you can see the structures that actually generate the alveoli, and you can see the critical role of the type 2 pneumocytes, and actually because of the tropism of the virus to angiotensin receptor 2, this is a receptor that actually the virus binds to. Because of the high expression of this receptor in type 2 pneumocytes, they become very tropic to the virus, and this is what is causing actually eventually in the severe cases of ARDS, the collapse of the alveoli and the disruption of the normal function of the lungs. So in the next slide, you can see a very complex picture that, of course, I will not go into details, but this is just to show the complex events that occurs when the innate immune system is recognizing RNA that is coming from a virus. Actually, we have specific receptors that we call pattern recognizing or recognition receptors, that they are responsible actually to sense RNA, viral RNA, and actually provide the outline for the defense against the virus. So basically, they generate a very robust response in different tissues that actually brings about recruitment of cells and other factors, that these are the cytokine factors that actually are responsible for the innate response and the first line of defense against the virus. You can see on the bottom part, in the B part of the slide, that actually transcription factors that we all recognize, like STAT1, STAT2, and others, are actually get into function or are being activated upon the induction of these pattern recognition receptors and all the signal transduction, including factors that are related to the NF-kappa-B signaling cascade, that actually eventually they bring about the induction of all these inflammatory cytokines, including IL-1, IL-6, IL-10, TNF-alpha, and others. What you can see also, if you pay attention to the red part of the slide, these red squares actually resemble viral proteins that serves as antagonists to the normal function of the innate response. So we have actually two parallel interaction or two parallel contradicting activities. One is a normal activity of the innate immune system to generate antiviral responses, but viral proteins that try to actually antagonize this interaction by generating protein-protein interactions with critical factors, for example, the I-kappa-B alpha subunit that is critical for the activity of NF-kappa-B, and this way the virus is trying to evade the innate immune response. So there are basically two pattern recognition systems. One is called the RLR family. This is a family of receptors that recognize RNA. And in the second slide, there is another system that is called the toll-like receptor system, in the next slide, which is actually another system that helps the innate immune response to generate the response. And the difference is which transcription factor are involved. In the previous system, the STAT family is more involved in the toll-like receptors. The NF-kappa-B system is more involved. So at this point, it's just the general knowledge about the system. Now, what I think is interesting to know, and this is I found it in some reviews, is that when people investigated the SARS, the original SARS coronavirus infections, they did experiments in macaques, and they found significant differences between young and old macaques that actually may implicate for the differences that we see in the pathogenesis of the disease and the likelihood that adult people will develop ARDS compared to young people. And what they found is that the transcriptional regime, or the level, the potency of the activation of transcription factors such as NF-kappa-B was much elevated in elderly people than in young people. In other words, for some reasons that people don't understand yet, the transcriptional response or the inflammatory response in elderly people was much more elevated compared to the mild response, probably the normal response that should occur in young people. And we know that the difference between mild response and high response of the immune system is associated with many pathological responses. In autoimmunity, in inflammatory responses, in atherosclerosis, in many, many diseases, actually diseases of the 21st century, the major difference between people that suffer this multifactorial disease and those that have less severe symptoms is the level of the basal activation of this innate immune system, because this activation of the immune system, of the innate system, will also dictate what is happening and what is the level of the activation of the adaptive immune system. So in the next slide, well, we can skip it because, again, it's the basis of... okay, now we come to the adaptive immunity. So following the initial stages of innate immunity, cytokines, cells, and other factors are actually activating 2 or 3 types of cells. They activate T cells, which are also differentiated into CD4 and CD8 T cells, and they activate B lymphocytes. The CD4 T cells secrete cytokines, which actually help 2 types of cells, the B cells, in order to differentiate into plasma cells and produce antibody response, and they help CD8 T cells to mature and become killer cells. This is why we call them CD4 helper cells. There are many subpopulations of this, but at the moment this is sufficient. So now, after the response has been generated, you have 2 kinds of protection. One is an antibody response protection. The antibody can bind the virus. If the virus has good epitopes, the antibodies can neutralize the virus. They can mark the virus for destruction by other immune cells like macrophages, and actually the antibodies can even prevent the virus to attach to the receptor if the antibodies are, of course, directed against epitopes that can neutralize or can block the attachment of the virus to the cell surface receptors. The cellular response in the form of CD8 T cells, these cells recognize peptides that are presented on MHC molecules on the surface of infected cells, and basically the CD8 T cells will eventually kill infected cells and will clear the infected cells within a week or 10 days. Okay, so this is a normal activity of the immune response, and this is the response that probably young and everybody that is infected with coronavirus 19 has such a normal immune system that at later stage is protecting. And indeed, there is evidence from the literature that immunity in the form of antibodies is protecting 90% of the people that were infected with coronavirus 19. So everybody that is infected is likely to resist another infection. This is data that is coming from a research lab just in the recent days, also from the lab in China. There is a recent paper that was published a month ago that from the lab that actually started to, the first labs that started to map the virus, to sequence and to study the immunology against the virus in this area of China. And they reported, first of all, that the protease and the receptor of the COVID-19 is similar to the SARS, and that people that have been infected and recovered, the antibodies, the sera from this patient, actually neutralized the virus in vitro. So in the next slide, so we can skip this one, Debbie. Okay, so in really 5 minutes, because Debbie needs to teach. So the development of vaccine is not a simple task as
Yoram,
yes?
if it can be in 3 minutes, because I need to sum up and then teach.
Yes. Thanks. So if we need to summarize the story of vaccine is not an easy task, like described in the media. There are many ways, many mode of actions to develop vaccines. The simplest mode of action is to develop antibodies. If you can go to the next slide. And the next slide. Okay, so what you see is a spike protein. This is actually the target that probably many labs are working at the moment to try to develop antibodies. This is the protein that actually is responsible for the infection. So this protein is responsible for attachment of the virus to the cells that are being infected. And of course, people are trying to generate antibodies against this protein. Just as a clue, antibodies to this protein can do two things. They can block the attachment of the virus, but they can also work actually to enhance by mechanisms that are not fully understood. It's probably related to the conformation of the protein. The protein can assume two conformations, one before it infects and a second after infection. And probably the conformation by which the antibody is trapping the protein is critical, but there is evidence that if the antibodies bind a conformation that is not suitable, actually the antibody can enhance the infection of the virus, can enhance the tropism and the binding of this receptor, this virus to the receptor. So this is just a simple example how development of vaccine is very, I don't know if slow, but should be taken very cautiously. And if you go to the next slide, you can just see on the next slide that a proper vaccine with all the clinical trials, and I'm not talking about identifying the most appropriate epitope, but you can see that it takes years. So on the best case scenario, it takes between 6 months to 1 year to develop the research days of finding the right epitope, and then additional 2 to 3 years to produce it, to go through all the phases until approval. So a vaccine that will solve now the problem of this outbreak is actually not realistic, and it raises a lot of questions of future vaccine development in this field.
Thanks very much, Yoram. I think it's a good tag team there.
Yeah.
I think I'm going to probably just, So this slide was just to really show people what's being done to test. You can test two ways. You can test by RT-PCR to look for actual particles with RNA in them in people, which says that there's an active infection. Potentially they could still be infective and shed. And an antibody-antigen assay that can tell you if somebody has already been exposed and has immunity to it, with the hope that perhaps measuring high levels of IgG and low levels of IgM will indicate that this person has passed, but together with the RT-PCR will even more so, and maybe those people can be released to go back to work and go back to a normal life, go into hospitals, etc., etc. Just briefly. Okay, so now I just want to wrap up. So I'll summarize to a certain extent. This is an emerging disease. It's jumped from animals, from bats probably, into a naive human population. It's highly transmissible, very relevant for this particular disease prior to and perhaps even without development of symptoms. And it's most dangerous for people with pre-existing conditions and the elderly. It's an enveloped virus, so it's sensitive to many disinfectants and decays in the environment, and it's effective. Alcohol is effective against it. Soap is effective against it. It's an RNA virus with a large genome. A large genomed RNA virus is normally thought to mutate wildly. This particular family of viruses has a proofreading ability, which means that it has less errors, less mutations. So perhaps we'll be lucky once we have a vaccine that it will be good for a period of time longer than the normal influenza vaccines. Estimates, I didn't go through these, but the estimates at the best and worst scenario, if we don't mitigate dramatically, range between 13,500 and 108,000 people dying. I can go through the calculation. It's basically based on 0.3% case fatality rate to 4% of, if we assume that 50% of the population is infected in this period. The current thought is it's probably more along the lines of 1%. We're talking about 45,000 people. But if we manage to keep this in check, then we could dramatically reduce this, as at this stage at least Germany is managing to do. Okay, so that's the end of this very long seminar.